Cell-to-cell adhesion. Cell-to-cell adhesion is a selective process where cells adhere to one another of specific types. This process is selective because cells from one tissue form, such as the heart or the tissue or the muscles, cannot adhere to cells from another tissue form, such as brain or any other form. The adhesion of like cells leads to the formation of many tissues, and this adhesion is mediated by cell surface proteins or cell adhesion molecules, also known as CAM. Adhesion between like cells is known as homophilic adhesion, while adhesion between different types of cells is known as heterophilic adhesion. CAMs. There are five main classes of CAMs: cadherins, immunoglobulin superfamily, selectin, mucins, and integrins. Adhesion using cadherins and selectins both require calcium ions, whereas the others do not. In this presentation, we're only going to focus on cadherin and integrins. Cadherins are a family of calcium-dependent CAMs and play a crucial role in cell-to-cell adhesion and tissue differentiation. There are over 40 different types of cadherins, but the most expressed or the most commonly expressed are E, P, and N cadherins. Cadherins are type 1 integral membrane glycoprotein, and they are made up of an N-terminal extracellular region which contains sites for calcium binding, a single transmembrane segment, and a C-terminal cytoplasmic tail. As mentioned earlier, there are over 40 different types of cadherins, but we are only going to focus on E, P, and N because those are the most commonly expressed. E cadherins are present on epithelial cells, but is also expressed in parts of the brain. They are usually present in cadherin junctions in mature epithelial cells, where they connect a cortical actin cytoskeleton of the cells that they hold together. Also, E cadherins are the first cadherins expressed during mammalian development. When mouse embryos that are deficient in the E cadherin fall apart or try to develop, they're not going to be implanted into this placenta. And if they don't get implanted, then there will be no development of the fetus or the embryo. So they play a very crucial role in development. P cadherins are found on cells in the placentarian epidermis. This also indicates that cadherins are very important in fertilization and stuff like that, because if the placenta doesn't fully develop, or if P cadherins are deficient and they don't help in putting the cells of the placenta together, then the fetus is not going to get enough fluid or enough nutrients from the mother, and it's going to die. The last kind of cadherin I'm going to talk about is the E cadherin, which mediates the homophilic interaction and holds most epithelial sheets together. The N cadherins are found on nerve, muscle, and lens cells. Calcium-dependent cell-cell adhesion. How does calcium binding lead to cell-to-cell adhesion? When calcium binds to the cadherin repeats located at the N-terminal of the cadherin molecule, it rigidifies the cadherin molecule and exposes residues that form the dimer interface. The head-to-head contact between cadherin dimers and adjacent cell membranes result in cell adhesion. So when the calcium binds right here, you see it makes it become rigid. And if two cadherins from adjacent cells become rigid, then they bind to each other, forming the cell-to-cell adhesion. The next type of CAM that we're going to talk about is integrin. Integrins are adhesion receptor glycoproteins. They combine to the extracellular matrix of adhesion molecules Integrins are the principal receptor in animal cells for binding extracellular matrix proteins, and they bind their ligands with low affinity to prevent cells from becoming irreversibly glued to the matrix and unable to move. An integrin molecule is made up of two non-covalently bound transmembrane glycoprotein subunits called the alpha and beta subunit. The same integrin molecule in different cell types can have different ligand binding specificities. The divalent cation binding domain are used to bind either calcium or magnesium. This determines both the affinity and the specificity of the binding of any integrin to its ligand. The type and location of an integrin molecule is determined by the beta subunit which it has. Beta 1 subunits are found in almost all vertebrate cells. The beta 2 subunits are expressed exclusively on the surface of white blood cells and help in fighting infections. There are at least 8 known beta subunits in humans, but the most important are the beta 1 and beta 3. The beta 1 subunit is important for implantation, and mice cells that lack the beta 1 integrin die at implantation. Beta 3 subunits are found on platelets and a huge variety of cells. Platelets interact with fibrinogen during blood clotting to prevent excessive bleeding. So if you don't have any beta 3 platelets, or I'm sorry, if you don't have any beta 3 subunits binding to the platelets or binding to the fibrinogen, then you're going to have excessive bleeding and that might lead to death. Focal adhesion kinases. When integrins cluster at the sites of cell-cell matrix contact, they form focal adhesions. Focal adhesion kinases are recruited to focal adhesions by intracellular anchor proteins which bind to the beta subunit of an integrin. The clustering of these FAK molecules causes them to phosphorylate each other on a tyrosine, thus creating a phosphotyrosine docking site for members of the SRC family. This SRC family kinases then further phosphorylate FAK, creating docking sites for the variety of intracellular proteins such as the small GTPase activation protein Rho. Rho is activated by the phosphorylation and activation of the Rho guanine nucleotide exchange factor Rho GEF, which indirectly regulates myosin light chain phosphorylation through the Rho associated kinase, also known as ROCK phosphorylation of the MLC phosphatase. This leads to more MLC kinase activity and thus cell migration.